Hello and welcome. My name is Paul Jennings. I'm an assistant professor at the Division of Physiology in the Medical University of Innsbruck, and today I'd like to present the molecular basis of genetic renal diseases. The kidney is responsible for whole body homeostasis. It receives about 20% of the cardiac output, meaning that nearly 1,500 litres per day are perfused per the kidney, and of this 180 litres are filtered through the kidney, producing about 1 to 2 litres of urine per day. Through this system, water, solutes including glucose, ions, amino acids, and proteins and vitamins are reabsorbed. Excess nutrients and waste products and xenobiotics are eliminated. In addition, the kidney is extremely important in the regulation of the acid-base balance and the regulation of vitamin D and erythropoietin. The functional unit of the kidney is the nephron, and here I've drawn a cartoon of this where the blood leaves the endothelial cells into the glomerulus, and then from the glomerulus into the proximal tubule, into the thin descending limb, the thick ascending limb, the distal tubule, and then the collecting duct. Each region depicted here has a different set of transporters and proteins, which means that each region handles glomerular filtrate differently. The kidney, unlike for example the liver, has a limited regenerative capacity. As I said, we start off with approximately 1.5 million nephrons per kidney. However, nephrons are lost throughout life due to processes such as oxidative damage, and there is no de novo nephrogenesis after birth. There's also little substantial evidence for the existence of adult renal progenitor cells. Thus, repair seems to be intrinsic to the nephron itself and limited. The functional capacity of the kidney is several times more than required, which poses a problem in that whole body homeostasis is not compromised until the majority of nephrons are lost, and by that stage it's usually too late to do anything about it. Chronic kidney disease is a progressive disease which terminates in end-stage renal disease that will require dialysis or kidney transplantation for survival. CKD is an aging disease primarily, but it is also aggravated and accelerated by extrarenal factors such as high blood pressure, diabetes, and cardiovascular diseases. In addition, chemicals, drugs, and environmental pollutants can initiate kidney disease and accelerate the CKD progression. CKD is a major societal and economic burden, with an estimated 10% of people being affected worldwide, and prevalence is predicted to increase dramatically. In addition to these factors, mutations in genes interfering directly or indirectly with renal transport processes can cause early onset renal disease, or they may cause homeostatic imbalances leading to diseases that are not rooted in the kidney itself. Here's a sketch of the location of mutations affecting renal function and whole body homeostasis. And these are the diseases I'm going to talk about in this lecture. So there's quite a few mutations found in specific proteins of the glomerulus, the proximal tubule, which is very important for constitutive reabsorption of water, amino acids, and glucose, has many associated genetic diseases. Then there's a few in the thick ascending limb, and one in the distal and one in the collecting duct that I will talk about. To be somewhat objective, I will use the online Mendelian Inheritance in Man database. So, according to their website, this is a comprehensive, authoritative compendium of human genes and genetic phenotypes that is freely available and updated daily. So, they have hundreds or maybe thousands of diseases with over 15,000 genes in. And what this database does is it focuses on the relationship between the phenotype and the genotype. And you have, for example, here with FJHN, which has a OMIM number of 16200. It gives you its name and the alternative titles, the phenotype genotype relationships, clinical symptoms, and phenotypic series. And it also gives you the publications that are associated with these observations. I will use as well the Human Protein Atlas, which is a nice compilation of normal tissue expression using specific antibodies in healthy tissue. And here, for example, just to illustrate, you can see this with this protein, which is gamma butyrobetane hydroxylase, that you can see on the left hand side that it's expressed almost exclusively in the kidney, but also a little bit in the liver and gallbladder, a little bit in the skin. And the right hand side you can see the protein expression score, which is expressed predominantly in the kidney and a little bit in the liver. And on the immunohistochemistry you can see there that B-box is expressed exclusively in the proximal tubular cells in a kidney tissue slice. So the first part of my talk will be focused to glomerular diseases, and so here I'd like to give a brief overview of the main players in the glomerular basement membrane and their functions. So the first line is the fenestrated endothelial cells. Then we have the basement membrane, which is composed of a composite of collagen 4, laminin, fibronectin, and heparin sulfate proteoglycans. Then we have the podocyte slit diaphragm, which is composed externally or between the foot processes of nephrin, and then internally of podocin and CD2AP. And together, the glomerular basement membrane excludes cells through the fenestrae, which are about 70 to 100 nanometer in diameter. Large proteins, so approximately over 40 kilodaltons, are excluded. Also proteins that have a diameter larger than about 3 nanometer are also excluded. And then proteins that are negatively charged are excluded. What comes through is basically everything except these. We could think of the glomerular filtrate as a plasma without or with very low protein. The first disease I would like to discuss is Alport syndrome. It has 3 genetic variants discussed, shown here by the OMIM numbers, and it's a disease of collagen 4 A5. In 1927, Cecil A. Alport reported a family of individuals which showed progressive renal disease accompanied by hematuria and deafness. He also observed that males died prematurely. We know now that Alport's disease is caused by mutations in the collagen 4A5 gene, which encodes the collagen type 4 alpha 5 chain. Collagen type 4 alpha 5 chain is a major structural component of basement membranes and one of the main components in the glomerular basement membrane. Mutations are X-linked, and the mutations discovered so far prevent collagen 4A5 from being expressed at all. and thus males suffer more from these mutations. In addition, uh, due to the intricacies of collagen formation, um, uh, the lack of collagen 4A5 expression may also lead to the degradation of unaffected or non-mutated uh, collagens. In a mouse model of X-linked Alport syndrome published by Ruelt et al. Uh, in 2004 in the Journal of American Society of Nephrology, we can get some hints why males are much worse affected than females. So in the left-hand panel, we can see that compared to the wild type, that in the male mutant mice, that there is no expression of collagen A5, but also there is no expression of collagen A3. In the female carriers, we can see that there's a weakened expression of collagen A5, but we can still see it in some of the tubuli and some of the glomeruli. And there's also expression of collagen A3, although weaker than the wild type. In the estimated survival function of the animals, we can see that mutant males die far before the carrier females, and the age of onset of death is a little bit before 20 weeks. Also, we can see that there is in the lower panel on the right hand side, we can see that there is a very elevated protein in the mutant males compared to the wild type males. And although there is proteinuria in the female carriers, this is a little bit attenuated compared to the males. In electron microscopy studies of humans with Alport syndrome, we can see different types of pathologies of the glomerular basement membrane, including in A, thickening and splitting, in B, an irregular thickness, and in C, thinning of the glomerular basement membrane. So it's possible that in Alport syndrome, the glomerular basement membrane actually starts off really quite thin, and then has a pathological accumulation of basement membrane proteins, and then has sections of both very thick and both very thin glomerular basement membrane, and then this leads to splitting of the glomerular basement membrane and a lack of a glomerular barrier at all. There are also two other types of Alport syndrome. While Z-linked Alport syndrome is the most common, which accounts for 85% of cases, there's mutations also in collagen 4A3 and collagen 4A4, which cause Alport syndrome, which have indistinguishable clinical symptoms. So the first is autosomal recessive Alport syndrome, which is caused by homozygous or compound heterozygous mutations in collagen 4A3 or collagen 4A4. And both of these genes are mapped to collagen or to chromosome 2. And then there's a very rare case of autosomal dominant Alport syndrome, which is caused by a heterozygous mutation in collagen 4A3. The clinical symptoms of Alport syndrome include deafness, which is seen to especially affect high frequencies. Then we have, as I said, thinning, thickening and splitting of the glomerular basement membrane. So the glomerular basement membrane is usually about 320 nanometers thick, and this has been reported to go as far as 1000 nanometers in Alport syndrome. And it's maybe that this extreme thickening can actually split the glomerular basement membrane. glomerular nephropathy is obviously seen, and this eventually leads to end-stage renal failure. Laboratory abnormalities include hematuria, which means blood in the urine, and also proteinuria. A second disease of the glomerular basement membrane is Pearson syndrome. Um, and the OMIM number is 609049, and this is due to mutations in laminin beta 2. Laminins are a major component of basal lamina of the basement membrane, where the epithelial or endothelial cells are attached to, and the laminins are secreted by the neighboring cells. They form heterotrimers made up of alpha, beta, and gamma chains, and they bind to other laminin molecules to form a sheet-like matrix. It is thought that the loss of laminin 2, or the inability of laminin 2 to form in these chains, could prevent either polymerization or secretion from the neighboring cells. And these two hypotheses were discussed in the quoted Jason paper of 2011, published by Chen et al. In the normal kidney, you can see laminin beta 2 expression in the basement membrane of the tubules, and also higher reactivity in the glomerulus. In Pearson syndrome, however, mutations in the laminin beta 2 gene, which is on chromosome 3, leads to decreased or even the absence of laminin beta 2 immunoreactivity in tissues. Pearson syndrome has a neonatal onset of nephrotic proteinuria, and end-stage renal disease typically develops within the first year of life. An investigation published in the Journal of Human Mutations in 2010 showed that there is even more severe types of Pearson syndrome, which cause a truncated laminin B2, which increases the onset of nephrotic proteinuria and increases the onset of end-stage renal disease. The clinical symptoms of Pearson's syndrome include effects on the eye, including non-reactive fixed narrowing of the pupil, aplasia or atrophy of the dilator pupillae muscle, hypoplasia of the iris and the ciliary body, and often blindness. Proteinuria and nephrotic syndrome are observations due to the renal phenotype. There is an early onset of end-stage renal disease. Death is usually in the first few weeks of life if dialysis is not given, and renal biopsy shows diffuse mesangial sclerosis. The final disease of the glomerular basement membrane that I will discuss today is glomerulopathy with fibronectin deposits 2, and it has an OMIM number of 601894. This disease is due to mutations in fibronectin, and here on the left you can see the proposed fibronectin structure. Fibronectin is a large dimeric protein of about 440 kilodaltons, consisting of two subunits, and both are encoded by the same gene, FN1, but several splice variants exist. There's two forms of fibronectin possible: the soluble fibronectin, which is a major component of blood plasma and is produced in the hepatocytes of the liver, and this is involved in wound healing and blood clotting. And then there's insoluble fibrillar or thread-like fibronectin, which is a major component of the extracellular matrix, and it's secreted by various resident cells, including fibroblasts. On the left-hand side, we can see a fibronectin dimer, and the different functional areas have different colors. So in blue, we can see the heparin binding domains. In red, we can see the cell surface binding domain, which contains this RGD motif, which is the arginine glycine aspartic acid sequence, and this is important for cell attachment via binding of integrin receptors. Then there's a fibrin binding domain and a collagen binding domain. GFND is caused by heterozygous mutations in the gene encoding fibronectin 1 on chromosome 2. Castelletti and colleagues have published a paper in 2008 in PNAS, which proposed that the disease may be due to impairment of fibronectin to correctly assemble into fibrils, causing an imbalance of the soluble and the insoluble fraction of fibronectin, and also that plasma fibronectin might be apparently incorporated into the glomerular matrix. The clinical symptoms of GFND include glomerular deposits which show immunoreactivity to fibronectin antibodies, nephrotic syndrome, enlarged glomeruli, and end-stage renal disease. Laboratory abnormalities include proteinuria and hematuria. The next set of diseases I want to discuss are diseases of the slit diaphragm. And just to introduce again the slit diaphragm in a little bit more detail. So we have on the top here you can see the fenestrated endothelial cells, and then again the basement membrane, which is approximately 320 nanometers thick. And then just below that we have the podocytes, which have these podocyte foot processes. And in between the podocyte foot processes are the slit diaphragms. There's a few components here that are important which are involved in these diseases. So there's the Wills tumor 1, which is a transcription factor resident in the nucleus of podocytes, and it's responsible for maintaining a podocyte-like phenotype. Then within the foot process, in the inside of the foot process, we have podocin CD2AP and Z01, and then the slit diaphragm itself, which is in between, in the extracellular space in between adjacent foot processes, is made up of nephrin. So the first disease of the slit diaphragms is nephrotic syndrome type 1, and this is due to mutations in nephrin itself. Nephrin is a 180 kilodalton transmembrane protein and is the main structural component of the podocyte slit diaphragm, as I said. It's a single transmembrane protein, and it has repetitive IgG domains that facilitate trans and cis interactions, forming a complete mesh between adjacent foot processes. So you can see here on the top left diagram, which is in 2 dimensions, you can see the blue nephrin, which is from the top foot process, and then the green nephrin, which is from the bottom foot process, and they meet at their IgG domains. And then if you can imagine this in a 3D structure on the right hand side, that uh, the, on the inside we have the endothelial cell, and then the blue is the glomerular basement membrane, and then we have the foot processes. Here you can see three different foot processes, and in the very bottom of the foot process you have this mesh of nephrons, and they protrude just slightly into the inside of the foot processes, and this mesh is all the way along the foot processes, forming a single diaphragm. On the left side here you can see the immunohistochemistry for nephron from Protein Atlas, and you can see that nephron is exclusively expressed within the glomerulus in the normal human kidney. The disease nephrotic syndrome type 1 is an autosomal recessive disease caused by homozygous or compound heterozygous mutations in the gene encoding nephron, NPHS1, on chromosome 19. It is characterized by prenatal onset of proteinuria, followed by severe steroid-resistant nephrotic syndrome apparent at birth with rapid progression to end-stage renal failure. Kidney transplant may be curative, although there is a risk of reoccurrence of nephrotic syndrome after transplantation. Here I've taken some results from Putala et al., which was published in 2001 in Human Molecular Genetics, and they showed in knockout animals, knockout mice, transmission electron microscopy, and here you can see quite nicely in panels A and B normal foot processes with nice slit diaphragms present in the magnification in B. And in C, in the knockout animal, you can see that there is a abnormal, irregular foot processes present. And in D, you can see that the slit diaphragms are basically absent, and there is an abnormal distance between the adjacent foot processes where they exist. And I also showed that the homozygous mouse had extremely high protein in the urine, which was not the case for the heterozygous or for the wild type animals. The clinical symptoms of NPHS1 include prenatal and postnatal symptoms. The prenatal symptoms include proteinuria, so this is excess protein in the amniotic fluid. Also increased alpha-fetoprotein in enlarged placenta, and often premature delivery. Postnatal, there exists nephrotic syndrome with severe proteinuria, dilated proximal tubules, tubular atrophy, interstitial fibrosis, mesangial cell proliferation, diffuse mesangial sclerosis, glomerular sclerosis and fibrosis, and loss of podocyte foot processes. The second disease of the slit diaphragms is the nephrotic syndrome type 2, and the OMIM number is 600995, and this is due to mutations in podocin. And here we can see the structure of the podocin homodimer. Podocin is a membrane protein of the band 7 stromatin family and has 383 amino acids. It is a transmembrane domain forming a hairpin structure with two cytoplasmic ends at the N and the C terminus, the latter of which interacts with the cytosolic tail of nephron, with CD2AP serving as an adapter. Podocin oligomerizes in lipid rafts together with nephron to form filtration slits. Here on the left you can see podocin attaching to the membrane, attaching to the plasma membrane, and then recruiting CD2AP and nephron, which then binds ZO1 and interacts with filamentous actin. On the left hand side here you can see immunohistochemistry uh, in a normal human tissue, also taken from Protein Atlas for podocin. And as with nephrin, you can see that it's only expressed in the glomerulus. The disease nephrotic syndrome type 2 is a autosomal recessive disease caused by homozygous or compound heterozygous mutations in the gene encoding podocin, which is called NPHS2, and this is on chromosome 1. The disease is characterized clinically by childhood onset of proteinuria, hypoalbuminuria, hyperlipidemia, and edema. Kidney biopsies show non-specific histological changes such as minimal change, focal segmental glomerular sclerosis, diffuse mesangial proliferation, and as with NPHS1, the disorder is resistant to steroid treatment. The clinical symptoms of NPHS type 2 are nephrotic syndrome, proteinuria, minimal change disease on renal biopsy, mesangial cell proliferation, focal segmental glomerular sclerosis, loss of podocyte foot processes, and mesangial IgM deposition. The third type of slit diaphragm disease that I will talk about today is nephrotic syndrome type 3. The OMIM number for this one is 607832, and this is due to mutations in the CD2AP gene. CD2AP, or CD2 associated protein, is a 70 kilodalton scaffolding molecule that regulates the actin cytoskeleton. It interacts with filamentous actin and a variety of cell membrane proteins through its multiple SH3 domains that you can see here on the bottom diagram, also through its proline-rich domain and through its actin binding sites. CD2AP is localized to membrane ruffles, lipid rafts, and the leading edges of cells, and is implicated in the dynamic actin remodeling and membrane trafficking that occurs during receptor endocytosis and cytokinesis. In the podocytes, it's required for slit diaphragm formation by anchoring podocin with nephrin, and you can see this here on the top left scheme. In the normal human kidney, you can see that CD2AP is located in pretty much all of the cells in the kidney, and this is the same in the rest of the body. So it's not exclusively located in the glomerulus. The disease nephrotic syndrome type 3 is conferred by a haploinsufficiency for CD2AP, which is encoded by the gene CD2AP on chromosome 6. So therefore there's too little or a total lack of CD2AP expression in the disease. The disease leads to focal segmental glomerular sclerosis associated with clinically with proteinuria, nephrotic syndrome, and progressive loss of renal function. It's commonly referred to in the literature also as focal segmental glomerular sclerosis 3. The final glomerular disease in this talk will be Dennis-Drash syndrome, OMIM 194080, and this is due to mutations in Wilms tumor protein. In the normal human kidney, you can see here in the protein atlas immunohistochemistry that Wilms tumor is Wilms tumor 1 protein is only expressed in the glomerulus, and there it's only expressed in the podocytes in the kidney. The disease Denise thrash is caused by mutations in WT1, which is expressed on chromosome 11. And these can be autosomal dominant mutations, or it's also been documented that somatic mutations uh, can can occur. The protein itself is a transcription factor, and it's important for the development of the urogenital system and also for the maintenance of podocyte differentiation. The clinical symptoms of Denise thrash include abnormalities of the male or female genitalia and severe kidney effects such as nephrotic syndrome, chronic end-stage renal failure, diffuse mesangial sclerosis, focal segmental glomerular sclerosis, and nephroblastoma can occur in the inherited disease or also due to somatic mutations. In the inherited disease, death usually ensues by the age of 3 due to renal failure. Now we'll move along to the proximal tubule, and there's a number of inherited proximal tubular diseases. And the first of these we will discuss are diseases of glucose reabsorption. Under normal conditions, all of the glucose is removed as it passes through the proximal tubule. This is achieved due to different apical and basolateral glucose transporters. The first of which is the sodium glucose transporter type 2, which is encoded by the LC582. This is a low affinity, high capacity transporter present on the apical side of the cells and is present in the S1 segment. The second transporter is the sodium glucose transporter type 1, which is a high affinity, low capacity transporter. It is not only responsible for glucose and sodium transport, but can also transport galactose, and this is present in the S3 segment of the proximal tubule, also on the apical side. Glucose that's brought into the proximal tubule is removed on the basolateral side through GLUT2, which is also a sodium glucose and galactose a co-transporter, and it's encoded by the SLC2A2. Now SGLT2 has recently become a target to try and alleviate the symptoms of diabetes by blocking this protein and allowing glucose and sodium to continue the journey through the rest of the nephron. So mutations in any one of these processes can cause renal glucosuria. The first of which is familial renal glucosuria, which is affected by mutations in SGLT2, which mean that the protein does not dock to the apical side or isn't present. The second is glucose-galactose malabsorption, which is due to mutations in SGLT1, which mean that the protein isn't present on the apical side of the proximal tubule and can't function. And the most severe of these is Fanconi-Bickel syndrome, which is due to mutations in the SLC2A2, the GLUT2 transporter. And if this isn't present, glucose is accumulated within the cell, causing glycogen accumulation and resulting in glycogen storage disease and toxicity. Renal glucosuria with the OMIM number of 233100 is caused by homozygous, compound heterozygous or heterozygous mutations in the sodium glucose transporter 2, SGLT2, which is encoded by the SLC5A2 gene on chromosome 16. So this is an autosomal dominant and a autosomal recessive disease. As you can see in the protein atlas immunohistochemistry, in the normal kidney, SGLT2 is present on the apical borders of the proximal tubular cells, and as I said before, in the mutation, the protein is no longer present in the membrane. So patients with familial renal glucosuria have a decreased renal tubular reabsorption of glucose, and thus glucose is found in the urine. However, there is no hyperglycemia, and there is no signs of other renal pathologies. So the only clinical symptom really is glucosuria, and there is rarely any significant health problems associated with renal glucosuria. Glucose-galactose malabsorption with the OMIM number of 606824 and is also abbreviated as GGM, is caused by homozygous mutations in the gene encoding the sodium glucose transporter 1, encoded by SLC5A1 on chromosome 22. This is an autosomal recessive disease, and it's not only due to the lack of SGLT1 in the kidney, but also the lack of SGLT1, which normally resides also in the duodenum, the small intestine, and the colon. And this is important in the first weeks of life, as lactose present in breast milk and infant formula is broken down into glucose and galactose in the intestinal tract, and the SGLT1 is responsive for the reabsorption of both. So the disorder manifests itself in the first weeks of life, and its GIT symptoms include diarrhea, which can be severe and chronic, and can be measured by increased bowel sounds. Also, there is mild glucosuria and metabolic effects such as hyperosmolar dehydration and metabolic acidosis. This disease can, if unnoticed and untreated, can be lethal, and a potential therapy is to eliminate glucose and galactose from the diet. The Fanconi-Bickel syndrome is caused by homozygous or compound heterozygous mutations in the GLUT2 gene on chromosome 3. This is an autosomal recessive disease, and it, as I've mentioned, causes glycogen build-up in the proximal tubule. It's an early onset disease resulting in poor appetite, galactose intolerance, and growth retardation. The clinical symptoms include glucosuria, generalized amino aciduria, and hyperphosphaturia. Metabolic effects include chronic acidosis and impaired galactose metabolism, and malabsorption in the gut. Potential treatment is replacing fructose for glucose, and to restrict galactose in the diet. The next set of proximal tubular diseases I want to discuss are the diseases that are involved in bicarbonate reabsorption. Under high concentrations of carbon dioxide and water will be metabolized to carbonic acid, which is then further metabolized to bicarbonate and protons. This situation occurs in generally within cells, and under low carbon dioxide tensions, the reverse happens. So the equilibrium goes in the opposite way, where bicarbonate and protons eventually end up in carbon dioxide and water. Now, in the proximal tubule, there is an apical carbonic anhydrase, which converts the protons and bicarbonate to CO2 and H2O, and H2O and CO2 can then diffuse through aquaportin 1 into the proximal tubule cell, and they're under the influence of carbonic anhydrase 2, which is the cytosolic form in all cells. The opposite happens, and bicarbonate and protons are produced again. Then the sodium and bicarbonate is removed through the SCLC4A4 sodium bicarbonate cotransporter at the basolateral side. Autosomal recessive mutations in the gene encoding carbonic anhydrase 2, CA2, on chromosome 8 cause this disease, osteopetrosis autosomal recessive 3, with an OMEM number of 259730, and the main symptoms of this are renal tubular acidosis. And mutations in the sodium bicarbonate cotransporter encoded by the gene SLC4A4 cause renal tubular acidosis proximal with ocular abnormalities and mental retardation with OMEM 604278. And the symptoms of this disease are renal tubular acidosis, bicarbonate wasting, and metabolic acidosis. The third set of proximal tubular diseases I want to discuss are diseases of receptor-mediated endocytosis. Under normal conditions in healthy individuals, all of the protein that is filtered through the glomerulus is recovered in the proximal tubule. This is mediated by the myelin-cubolin system, where proteins dock to myelin, initiating a conformational change, causing recruitment of ARH, and then attraction of clathrin, which cause the development of an endocytotic membrane and eventual endocytosis and the development of the early endosome. In the early endosome, CLIC5 is important for the recovery of myelin-cubolin back to the apical membrane. The early endosome progresses to the late endosome and finally to the lysosome, where the contents, which include proteins and vitamins, are expelled to the basolateral domain into the interstitium. Now, proteins can bind other molecules such as vitamins, and this is the main process also how essential vitamins such as vitamin D and vitamin A are recovered. In Donny-Barrow syndrome, also called facio-oculo-acoustico-renal syndrome, or 4FOAR for short, mutations in the LRP2 gene, which is the gene encoding myelin on chromosome 2, cause the disease. As you can see in the protein atlas immunohistochemistry, there is extensive staining in proximal tubular cells for myelin, and this is not the case in Donny-Barrow syndrome, and it's thought that loss of essential vitamins in the urine are responsible for the phenotype. Clinical symptoms of Donny-Barrow syndrome include disturbances of the face and nose, including a large anterior fontanelle, macrocephaly, mid-face hypoplasia, and typically low-set ears. The nose is typically short with a flat nasal bridge and a broad tip. In the kidney, proteinuria is observed, and especially retinal binding proteins and vitamin D binding proteins are found in the urine. Also, there can be disturbance of the eye, which may cause loss of vision, and deafness is also a possibility. Dent disease type 1 with the OMEM number of 300009 is caused by mutations in the chloride voltage gated channel 5 encoded by the gene CLCN5 on the X chromosome. So because it's an X-linked recessive disease, males are often more severely affected. And the mutations in CLCN5 cause an impaired recycling of megalin and cumulin in the proximal tubular cells. The clinical symptoms of Dent disease include severe kidney effects, including nephrocalcinosis, which is calcium salt deposition, kidney stone formation, renal insufficiency, and renal failure in adulthood. Laboratory abnormalities include low molecular weight proteinuria, hypercalciuria, hyperphosphaturia, amino acid urea, glycosuria, and microscopic hematuria. And often patients develop rickets in about 33% of all cases. There is a softening of bones, there's increased fractures, bone pain, and sparse bone trabeculae and tiny bone cortex. It is likely that many of these symptoms are due to a loss of vitamin D due to the inability of vitamin D to be taken up in the proximal tubule. In the final set of proximal tubular diseases, I want to discuss are diseases that affect amino acid handling. In this cartoon, which I've adapted from a publication by Verrey et al., you can see the main processes involved in proximal tubule handling of amino acids. On the luminal side, which is on the top right hand side, you can see that the sodium neutral amino acid cotransporter is comprised of two proteins, in blue BAT1 and in green ERBAT. These are encoded by the gene SLC7A9 and SLC3A1, and they're responsible for the transport of cysteine and cationic amino acids from the lumen into the proximal tubular cell. Mutations in either one of these genes causes cysteinuria, and if cysteine rises above 1 millimolar in low pH environments, stone formation can happen, and that's why under certain conditions acetazolamide is given, which increases the luminal pH and prevents stone formation. On the basolateral side, we can see that cationic amino acids are transferred to the interstitial space via this complex of Y plus LAT1 and 4F2HC. Mutations in the gene that encodes Y plus LAT1, which is SLC7A7, cause lysinuric protein intolerance, and I'll explain these two diseases in a bit more detail in the next two slides. Cysteinuria with an OMIM number of 220100, as I said, is caused by either mutations in the SLC3A1 amino acid transporter or the SLC7A9 transporter. There are both recessive and dominant forms, and onset is usually in the first or second decade. Renal features include stone formation and renal failure, and laboratory abnormalities include increased urinary cysteine and increased urinary lysine, arginine, and ornithine. Treatment can be, as I said, by attempting to increase the pH of the luminal fluid by giving, for example, acetazolamide, which is a carbonic anhydrase inhibitor. Lysinuric protein intolerance with an OMIM of 222700 is caused by homozygous or compound heterozygous mutations in the cationic amino acid transporter gene SLC7A7, which is on chromosome 14, and as I said, encodes the Y plus LAT1 protein. Deficiency of arginine and ornithine restricts the function of the urea cycle and leads to hyperammonia after protein-rich meals. This can cause chronic kidney disease, increase in cationic amino acids, urinary lysine, arginine, and ornithine. And in the gut, this can manifest as nausea, vomiting, diarrhea, aversion to protein-rich food, and impaired intestinal absorption of cationic amino acids. Hepatomegalia, alveolar proteinosis, pancreatitis, and splenomegalia can also occur in lysine-uric protein intolerance. Next we move to mutations of the thick ascending limb. The first disease I want to discuss is familial juvenile hyperuricemic nephropathy, FJHN for short, and this has an OMIM of 162,000. On the left hand side is depicted the expression of uromodulin in epithelial cells in vitro, which overexpressed the wild type uromodulin, and on the top panel is a top down view, and on the bottom panel is a Z stacking of this. So you can see that the wild type uromodulin is expressed mostly on the or exclusively on the apical side of cells in vitro, and this is the case also in vivo. So FJHN is caused by mutations in the uromodulin gene, which is also called Tam-Horsfeld protein, and the gene resides on chromosome 16. Uromodulin is exclusively expressed in the thick ascending limb in the kidney and is targeted to the apical side via GPI anchorage, and it's secreted at a rate of about 50 milligrams per day. So it makes it one of the few, if not only, proteins that are made to be secreted into the urine. Its functions include decreasing ascending bacteria invasion by binding to type 2 fimbriated E. coli. It decreases calcium oxalate stone formation, and it also acts as a pro-inflammatory signal for breach of tubular epithelium. FJHN and related uromodulin-associated diseases are caused by a delay in uromodulin ER processing and activation of the unfolded protein response. And we think this then causes eventually the death of the thick ascending limb cells. The main feature of FHN is hyperuricemia and gout, which then develops into severe renal side effects such as tubular atrophy, the thickening of the basement membrane, often small medullary cysts, and end-stage renal disease in the second or third decade. The hyperuricemia can be treated giving allopurinol, which is an antioxidase inhibitor, but this doesn't alleviate the renal symptoms. An early indicator of FJHN is a decrease of urinary uromodulin in the urine compared to healthy controls. The second thick ascending limb disease I want to discuss is Bartter syndrome. However, it's not just one disease, it's several different diseases which affect different transporters in the thick ascending limb. There's 5 types of Bartter syndrome, each one affecting different transporters in the thick ascending limb. The BARTS1 affects the apical sodium potassium chloride transporter. BARTS 2 affects the apical potassium extruder ROMPK. BART3 affects the CLC NKB, which is a basolateral voltage gated chloride channel. BART4 affects Barton, which is a modulatory protein of the CLC NKB. And BART5 affects the calcium sensing receptor, which seems to become overactive and is responsible then for suppressing ROMPK and Claudin 16, which is a paracellular tight junction protein responsible for the paracellular diffusion of calcium and magnesium back into the interstitial space. Bartter syndrome 1, 2 and 4 have antenatal effects affecting the fetus, while the BARTS 4 is the only one which is known to cause deafness. All of the Bartter syndrome cluster of diseases are characterized by low sodium and low potassium levels, increased blood pH causing metabolic alkalosis, and normal to low blood pressure. Similar effects can be seen with the therapeutic use of loop diuretics, for example, furosemide. Lab abnormalities include elevated renin and aldosterone, and renal symptoms include polyuria, which is excessive urine volume, polydipsia, which is excessive thirst, and increased urinary sodium, potassium and chloride. So we move on now to the distal and collecting duct diseases. And the first of this is Gittelman syndrome, which is caused by homozygous or compound heterozygous mutations in the SLC12A3 gene, which encodes the sodium chloride cotransporter NCC, and this is on chromosome 16. It's an autosomal recessive renal tubular salt wasting disorder characterized by hypokalemia, metabolic alkalosis with hypomagnesemia and hypocalciuria. It has an early onset in childhood, and the clinical symptoms include polyuria, renal potassium wasting, renal magnesium wasting, metabolic effects such as hypokalemic alkalosis, polydyspia, and lab abnormalities include hypokalemia, hypomagnesemia, hypocalciuria, and increased renin. One of the main regulatory roles of the collecting duct is vasopressin-dependent reabsorption of water, and the process is described here, where you normally have a hypo-osmolar luminal fluid and a hyper-osmolar interstitial fluid. And in the absence of vasopressin, aquaporin 2 is located in a vesicular body within the cells, and when vasopressin is present, which binds to the basolateral V2 receptors, there is a signaling cascade which increases cyclic AMP and then facilitates the aquaporin 2 vesicles to dock on the apical side, increasing the water permeability of the apical membrane and allowing water to come through. The water then exits on the basolateral side through aquaporin 3 and aquaporin 4 molecules or transporters. So there's two types of collecting duct diseases that I describe here. That's diabetes insipidus nephrogenic X-linked, which has an OMIM 304800, and this is caused by mutations in a gene encoding vasopressin V2 receptor. And there's the diabetes insipidus nephrogenic autosomal with an OMIM of 125800, and it's caused by heterozygous and homozygous or compound heterozygous mutations in a gene encoding aquaporin 2 water channel, which is mapped to chromosome 12. So, in summary, I've discussed the molecular basis of many of the genetically linked renal diseases, starting with the genetically linked glomerular diseases. So we had three diseases that were involved in the formation of the glomerular basement membrane. That was Alport's, which affects collagen 4, Pearson's, which affects laminin B2, and glomerulopathy with fibronectin deposits 2, which affects fibronectin. Then we had a cluster of diseases which are the steroid-resistant nephrotic syndromes, which affect the formation of the slit membrane. That's these diseases affect nephrin, podocin, or CD2AP expression. And then we have the Denise-Strash syndrome, which affects the expression of Wilms tumor gene. In the proximal tubule diseases, we discussed diseases of glucose reabsorption affecting the SGLT2, the SGLT1, and the GLUT2. Diseases of bicarbonate reabsorption, which affect carbonic anhydrase 2, and the sodium bicarbonate cotransporter. Diseases of receptor-mediated endocytosis, including Donny-Barrow syndrome, which affects megalin, and the Dent disease 1, which affects CLCN5, and diseases of amino acid handling, including cystinuria, which affects ERBAT, and lysin-uric protein intolerance, which affects the Y plus LAT1 on the basolateral membrane of the proximal tubule. In addition, we discussed diseases of the thick ascending limb, including juvenile hyperuricemic nephropathy, which affects the protein neuromodulin, Bartter syndrome, which affects several proteins, including NKCC2, ROMPK, CLC, NKB, Barton, and CSA-SR. And we quickly talked about genes that affect the distal tubule, including Gittelman syndrome, which affects the NCC protein, diabetes insipidus nephrogenic X-linked, and diabetes insipidus nephrogenic autosomal, which affects vasopressin V2 receptor and aquaporin 2 respectively. Today we have discussed a whole host of hereditary mutations leading to renal diseases with specific clinical symptoms. The study of these diseases allows us to better understand the molecular biology of renal function and the roles of different proteins in glomerular filtration and renal transport. Also, by better understanding these diseases, it may be possible to develop avenues to alleviate symptoms or even to develop new therapeutic strategies. Thank you for your attention. Goodbye.